What you see here on the screen, and first of all, can you hear me? Can you hear me?
Yeah.
Okay.
Yes, we can, Scott.
Okay, thank you. Okay, first, what you see here on the screen is the river from Conrad Hal Waddington's book, Organizers and Genes, and kind of an aerial view of this river. And I want to talk about three rivers: metabolism, development, and evolution. But first, I want to thank you, John and Sabina, for having me here on Zoom. It's a joy and an honor to be here. John, I've been retired for a few years now, and what I learned is that there's a Spanish word for retiree, male retiree, which is far better than pensioner or retiree. It's jubilato, that we are those who rejoice. So jubilate. So the first thing I'm going to talk about is just going with the flow of this river and how I kind of got into this crazy notion. And as John is often saying, no man ever steps into the same river twice. It's not the same river, nor is he the same man. And so one should celebrate one's development, one's metabolism, one's metamorphosis. And there are these rivers within rivers. There's, of course, the great river, the flow of evolution, the flux of genes from one species to another. And then there's, of course, the flow of development, the life cycle from gamete formation, fertilization, cleavage, gastrulation, adulthood, and so forth. And then within that, there's the flow of metabolism, this magical flow that allows us to retain our identity by changing our parts. So that there are these three flows that I want to talk about and that I want to kind of unify. So within flows, there are rivers within rivers, whirlpools within whirlpool. The whirlpool has often been used as a metaphor for life. Baron von Cuvier said the living being is a whirlpool, constantly turning in the same direction, in which matter is less essential than form. And of course, Dan Nicholson recently, the existence of a whirlpool, for instance, is a direct consequence of its own unremitting activity, which is what enables it to maintain itself through time. In the same way, if an organism is to stay alive, it has to keep acting, working in the thermodynamic sense to avoid the ever-present threat of equilibrium. And so here we have Hiroshige's Naruto whirlpool in 1853, and here's the same whirlpool last year, still there, still changing its parts to retain its identity. Now, I got into this crazy business of looking at organismal individuality and flux in 1977, when I was a postdoc at the University of Wisconsin. But being a postdoc was my day job. What I really was in the evening was a British sailor on the HMS Pinafore. There's a picture of me and one of my fellow tars. And he asked me, one of my fellow sailors asked me on the way to the rehearsal, why are there separate biochemistry and molecular biology buildings? What's the difference? And I gave the standard structure versus function difference between molecular bio and biochemistry. And as I was saying it, I disagreed with myself. So I actually went and tried to find the difference and eventually wrote a paper on this where I wrote, thus, by 1945, one finds two rival claimants to the studies of the physical basis of life: the older biochemical school, conducting its research with cells in the tradition of flux, and the aggressive molecular biologists working with viruses in the newly resurrected tradition of the crystal nature of life. And so we have the metabolic view, the replicative view, one dealing with flux, one with permanence, one looking at the metabolizing cell as the unit of life, one looking at the replicating virus as the unit of life, and here we have biochemistry and molecular biology, the reason for those two buildings at the University of Wisconsin campus. Now, oops, okay. So I was overjoyed when I saw in 2009 this wonderful paper by John Dupre and Dupré and O'Malley looking at what is a biological individual. Our central argument is that life arises when lineage-forming entities, that's the replication, collaborate in metabolism, it's the flux. By conceiving of metabolism as a collaborative process performed by functional wholes, which are associations of a variety of lineage-forming entities, we avoid the standard tension between reproduction and metabolism in discussions of life. And that I thought was just such a critical thing. I had argued in my paper that DNA synthesis, where an enzyme took instructions from its template, and the operon model, where the differential synthesis of enzymes occurred at a molecular level, were integrative notions of biochemistry and molecular biology. But here we see a whole new area, this notion of metabolic pathways between organisms. Yes. Okay, so the first thing I want to discuss is this holobiont perspective on physiology. And I'll use here a very general definition of holobiont, that a holobiont is an assemblage of a host, just a big symbiont, and many other species living on it or in it, which together form a discrete physiological, developmental, and immunological unit through symbiosis. And one of the things that Dupré and O'Malley showed was that there was a metabolic cooperation between these entities. And indeed, that is what we see. And this is one of my favorite. This is the insect Planococcus, the mealybug. And to make phenylalanine, you have to use three organisms. There's Planococcus, which is the insect. You have Tremblia, which is a bacterium. And you have Morinella, which is a bacterium living inside that bacterium. And so to get the pathway for phenylalanine, an essential amino acid, you go from the substrates in the bacterium, processing it through substrates in the symbiont, back into the symbiont again, into the bacterium. And the last step is done by the actual insect's enzyme coded by the insect's genome. One has here genomic and physiological coordination because the various bacteria have lost some genes. They don't need these genes. Similarly, the insect has lost some genes. It doesn't need them. These are now kind of functional units making together this holobiont organism. In 2013, Margaret McFall-Yai and others, like 25 others, made a synthesis, wrote a paper in PNAS, Animals in a Bacterial World, a new imperative for the life sciences. Now, we don't see that word imperative often in a scientific title, but this was what it was. It basically says you cannot understand the life sciences unless you understand bacteria. And bacteria, it was argued, are part of us. They are necessary for our digestive functions, our neuroendocrine functions, circulatory, immune functions. They're necessary for coordinated bone growth, that the microbes are putting forth metabolites that either work as signals or work as substrates for the host body. As much as 1/3 of the animal's metabolome, the diversity of molecules carried in the bloods, have a microbial origin in mammals. Thus, the circulatory system extends the chemical impact of the microbiota throughout the body. 1/3 of those small molecules in our blood are derived or induced by microbes. The same year, Smith et al. And Jeffrey Gordon's lab published a paper on kwashiorkor, showing that it was not only a disease of protein deficiency, it was a disease of microbial dysbiosis, having the wrong microbes in one's gut. And they coined the word co-metabolism. microbe-host co-metabolism as a function of microbiota and host diet. So we have here this notion that there's a physiological connection, as O'Malley and Dupré said, between the parts of a holobiont. Now, I want to now talk, getting away from the physiology to development. And I want to talk about gut capillaries and pancreatic beta cells in mammals and sponge metamorphosis briefly. So development, I think we've seen, is actually a team sport, whereas symbiosis is usually described really as between consenting adults. We have something which is deeper. We have that the adult is made by the symbionts, process sometimes called sympoiesis. So on the left here are the intestinal vessels of a germ-free mouse. These mice do not have microbes in their gut, and they have a very poor circulatory system shown in green here on the villi of their intestine. You add the microbes back, and you get a great capillary network, which can take the nutrients from the intestine to the rest of the body. So here we have what the microbes can do. They can help make the capillary network of the mammalian intestine. How do they do it? They do it by an amazing way. They actually induce gene expression in the mammalian cells. Just as embryonic cells induce gene expression in each other, so the microbial cells are inducing gene expression, in this case, in the Paneth cells of the mouse intestine. And it's many genes that they're inducing. They're inducing SPRR2A, which is a matrix protein, extracellular matrix protein, a huge relative RNA abundance in SPRR2A. Colipase, an enzyme involved in lipid metabolism. Again, here we have the crypt cells. Here's germ-free. Here's what you get with the microbes added. And angiogenin, this is now known as angiogenin 4, was in the original paper, is angiogenin 3. In the epithelium, here's the germ-free over here. Here is the mRNA in the animals once they get the microbes. And here's, again, looking at the angiogenin 4, fascinating. You have here, this is set to 100%, 1.0. You have about 10-fold more angiogenin 4 mRNA in the mice with the microbes than you have without the microbes. So here is the conventionally raised on the red. Here you have 10-fold as much as in germ-free. Germ-free mice are like mice with a mutation that only produces 10% of the angiogenin 4 RNA. Most of this is due to Bacteroides theta iota micron. You get back to normal levels when you just add that to the host. And E. coli will do it a bit. B. infantis will do it a bit. And here is when you add the gut bacteria back. Here's where you have the conventionally raised. So E. coli and B. infantis can do it, but not as good as Bacteroides theta iota micron. And so as Ford Doolittle said, you know, it's not the singer, it's the song. I want to continue with the 1970s song titles and go, but nobody does it better than B theta iota micron. Others can do it. There's some that do it better. So angiogenin 4 and beta theta iota micron help organize the gut microbiome and the gut epithelium. Angiogenin 4 induces capillary formation. Angiogenin 4 expands the intestinal stem cells in the crypt. Angiogenin 4 also, and this is really neat, is toxic to Listeria, Candida, and Salmonella, which are potential competitors of Bacteroides, as well as being pathogens for us. So basically, you have an immune system which is comprised of both host and microbial functions. Bacteroides theta iota micron also secretes a DNase to form bile-dependent biofilms. So it's helping organize the gut microbes, both in terms of population and in their ability to stay within the intestine. So host and symbiont work together to structure the intestine and its microbial community. Immunity, I'm not going to go into it now, is a holobiont, not a host property. The conserved bacterial protein BEF-A expands pancreatic beta cell during zebrafish development. So Aromonas veronii, which is a microbe which is very rare in the seawater, or the river water for that matter, is needed for the pancreas to expand its beta cells. So again, here is a germ-free zebrafish pancreas. The green cells here are the insulin-secreting beta cells. Green is insulin. Here's conventionally raised, much more of the beta cells. But if you take a germ-free zebrafish rather, and you add into its gut this BEF-A protein, you can expand that population of beta cells. That one gene, BEF-A, is different between those microbes that will allow the expansion of the beta cells and that which don't allow the expansion of the beta cells. BEF-A protein causes division from pre-existing beta cells. So you have a protein, BEF-A protein, which is made by a microbe, and it corrects beta cell paucity in sterile zebrafish and in germ-free mice. There is a human BEF-A analog in Klebsiella, and it can rescue germ-free zebrafish beta cell mass. As you know, there's this hygiene hypothesis which says perhaps we're getting rid of the trace bacteria that are needed for normal development and normal health, and this may be a good, kind of case in point in zebrafish, that you have here a bacteria which is needed in trace amounts to cause the production of BEF-A protein, which will induce the normal expansion of beta cells. We see here the symbiotic linkage of those developmental and nutrient streams. Now, I want to talk about the convergence of these three streams of evolution, metabolism, and development. So we have here this bug, this bean bug Riptortis. Riptortis is an agricultural pest. It eats beans. It eats soybeans. And Riptortis nymphs, those bugs in the ground in their second instar, acquire a bacteria, Burkholderia, and it needs to be acquired anew each generation. It's not passed in the egg cytoplasm or anything. It has to be gotten from the soil. And Burkholderia is good to have because it promotes the larval bug growth, or the nymph bud growth, by stimulating juvenile hormone synthesis. So you get growth when you have this bacteria in the second instar. Now, the farmers spread this insecticide, phenanthrothione, on the beans to kill those nascent bugs. However, some Burkholderia have a metabolic pathway that allows it to use the phenanthrothione as food. And here's the pathway down here. You go to phenanthrothione to methylhydroquinone to basically a carbon source that can be used as a nutrient. So some Burkholderia can metabolize the pesticide, the insecticide, into food. The bugs that incorporate the pesticide-eating Burkholderia become resistant to the pesticide. So the bugs are giving them pesticide resistance. So Riptortis is being selected as a holobiont, even if Burkholderia can exist independently. We see this in other pests. The red turpentine beetle in the Pacific Northwest, where I live, is an innocuous, some people call it a pest, other people just say It's a degrader of dying trees such that it brings them back into the soil again. It's not even a pest. If it's a pest, it's a minor pest. It infests trees that have already been damaged. It can't deal with the immune system of a functioning, healthy tree. However, the beetle doesn't digest the wood. It's the fungi that are covering the beetles that digest the bark, make those holes, and give the beetles a home and a place to reproduce. Okay, and eventually they'll take the tree down and bring it back into the ground again. However, they were introduced into China, probably through railroad ties, and these beetles found other fungi, and these fungi were more aggressive, and they can digest healthy trees. They can digest wood better than the native fungus in the Pacific Northwest. Over 10 million trees have been destroyed in China by this beetle. It has become a major pest by changing its symbionts. Recently, the biochemical pathway of symbiotic bacteria was shown to be needed for the metamorphosis of sponges. So in the sponge life cycle, you can go from the adult that makes gametes, the sperm and the egg combine to make a larva, and eventually the larva, when it gets cues from coralline algae, will settle and metamorphose into an adult. Now, to do this, though, it needs a signal, which is nitric oxide. Where is it getting its nitric oxide? It's getting it from a bacteria. The arginine that is used for making the nitric oxide by the sponge's own nitric oxide synthase is made by the symbiotic bacteria. So this symbiotic bacteria, which is vertically inherited, it goes through the eggs of the gametes. So this eggs of the adult, this bacteria makes the arginine substrate, which is necessary for the metamorphosis of its host. So again, this combination of metamorphosis, the life cycle, with the, in this case, metabolism of the bacteria. Now, dung beetles are amazing, and Armin Moksick's lab has done amazing work uniting metabolism, development, and evolution. And here we see a female dung beetle rolling food, dung, on which its eggs will be laid. The dung beetle will make a hole and lay its eggs on that dung mass. Now, that dung mass is important. If you are a male, your behaviors and your phenotype will depend on the quality and quantity of that dung that your mother left you as a gift. Now, if you have poor dung or not much dung, you get very small horns, if any. If you have a lot of good quality dung, you get these kind of samurai helmet dung beetle horns here. So the amount and quantity and quality of dung is important. The horns are sigmoidally allometric with increasing body size. The beetles metamorphosing at large body size have horns of different sizes, and the more nutrition you get, the bigger the horn is. But below a threshold, males are hornless. And then there's this intermediate stage here, and the set points of when you get the horns and when you don't get the horns differ between species. The set points are different, and you can see these set points here, different sizes, different species. Okay. There are even different set points within a species. So in this species of dung beetles, depending on where it is, whether it's in Florida or Michigan, you get a different set point as to when you start forming horns. Okay, so the set points are different. They can be genetically fixed, or they can be environmentally plastic. The presence or absence of horns determines male reproductive strategy in some dung beetle species. So here you have the female, and the female has made her brood ball. This is the dung, and here's an egg on the brood ball, and the egg was fertilized. Now, it could be fertilized by this male over here with the big horns. That's the one who's guarding the gates here so that no other male comes in. And it will fight with other horned males. And the ones with the bigger horns generally win the battle. However, if you don't have horns, what are you going to do? Well, then you could become a sneaker male, because if you don't have horns, you can build tunnels, and you can make a tunnel that intersects with the female's tunnel, and you can mate with the female and fertilize the egg that becomes part of the brood ball. And about 50% of those are from sneaker males. Now, the dung beetle life cycle depends on that dung ball. The egg is placed on a maternally derived pedestal inside the dung ball. That pedestal on which the egg is placed has on it, or in it, fecal matter from the mother. The mother puts fecal matter between it and the dung. And that fecal matter contains the symbionts that will digest the dung. The gut symbionts shape nutritional conditions experienced by the larvae as the larvae's own habits of eat, excrete, re-eat, and repeat influence brood ball consumption. So this larvae that gets out of here, that gets out of the egg, first meets with the symbionts, digests the symbionts, or brings the symbionts into it, and using the symbionts is able to digest all the stuff that the cow was not able to digest, put into its dung. And then after it eats the dung ball, it excretes. And then it eats what it excretes. And it goes on and on, eat, excrete, re-eat, repeat, so that eventually it gets big enough so that it becomes, the larva undergoes metamorphosis, and then it can mate, and the adult beetles undergo maturation depending on environmental circumstances, which includes the production of the brood balls and the pedestals that contain the microbes, which influence the developmental and environmental aspects experienced by the offspring. So we have here a life cycle which is critically dependent on those symbionts that were left by the mother dung beetle. There's even a coevolution of the symbionts and hosts, such that in one species, if you change the microbes with that of another species, very closely related species, you find that when you change the microbes and you give one species A the microbes of species B, you get low robustness of beetles with the symbionts of other species. The adult size goes down significantly. Survival rate and developmental time here are about the same. However, with this species B, if you give it the symbionts of species A, you get low survivorship. You do not get many of these insects surviving. So these Onthophagus dung beetles associate with non-interchangeable host species-specific microbial communities that have the potential to influence growth, development, and fitness of the host. Again, in addition to it's the song, not the singer, there's a nobody does it better aspect to this as well. Now, some unpublished data that was mentioned in this book is that the symbiotic and niche-constructing behaviors shield the genetic variants from selection. And so again, you bring in that third aspect, that third river. But to really bring together the three streams of metabolism, development, and evolution, fetch a lavash. You need the cow. This is kind of my bullshit lecture. I talk about the dung beetles and I talk about bovine metabolism. Okay. The cow is a machine for turning grass into milk. 1869. You have complex carbohydrates, the plant material, cellulose, being digested by bacteria into simple sugars. Those simple sugars are taken by bacteria and archaea as well into short-chain fatty acids, butyric acid, propionic acid, so forth, which then can be used for aerobic respiration, gluconeogenesis, lipogenesis. So the cattle is able to go from complex carbohydrates, which it's eating, to the short-chain fatty acids, which are necessary for its respiration, gluconeogenesis, and lipid formation. Now, the cow may seem to be a strange model system for looking at evolution, but actually it was one of the first. Sewell Wright got his start as a senior animal husbandman in the Animal Husbandry Division of the United States Department of Agriculture. And here is his book, The Principles of Livestock Breeding, where he talked about small populations and genetic drift, facilitating new gene combinations. New interactions can be used as a substrate for selection. He developed the inbreeding coefficient, and shifting balance was formulated for livestock improvement. The cattle is actually one of the first kind of evolutionary model systems. And the cow, being an herbivore, has to be looked at in this manner because herbivores are derived. The basal group of animals usually contain detritivores, filter feeders, carnivores, but not herbivores. Within the ecdysozoa, for instance, the plesiomorphic state is primarily carnivory. In vertebrates, predation was often the ancestral condition from which herbivory evolved. So we have this paper saying that if you look at the animal kingdom, what you find is that the herbivores came after the carnivores, that eating an animal is kind of easy. Eating plants are difficult. They have these cellulose, hemicellulose, pectin compounds that you have to have special enzymes for. And those enzymes are not coming from the genomes of the animals. They're coming from the genomes of the symbionts, primarily the bacteria. As I said, eating animals is easy. Eating plants is difficult. Here we have an early Permian caseid, which was basically a large fermentation vat on legs. Ruminants are probably one of the most successful of the mammalian clades. Ruminants are terrestrial herbivores, including important livestock species such as cows, buffalo, yak, sheep, goats, and reindeer. They are one of the most successful mammalian groups in the world, both numerous and widely distributed throughout diverse habitats. So you have all these really fascinating animals over here. They are ruminants, giraffes, cows, deer. They evolved when a new part of the digestive tract, the rumen, which is basically a storage house for microbes, originated about 35 million years ago from distal portions of the esophagus adjacent to the normal stomach. So here we have the rumen. Here's the rumen. Here's the omasum, abomasum, basically what our stomachs are. The rumen is essentially a large anaerobic fermentation chamber where plant-degrading rumen microbiota, bacteria, protozoa, archaea, and fungi ferment otherwise non-digestible plant-based foodstuffs into the primary volatile fatty acids, acetate, propionate, and butyric acid. Okay, so this is a huge part. It's like 85% the volume of an adult cow. And it's used for this nutritional symbiosis. Cows are obligate herbivores. There's no cow without the symbionts. It's an obligate holobiont. The rumen microbiome produces up to 70% of the energy that the cow needs. Priority effects explain functional convergence, that emphasis is mine, in the rumen microbial composition across the developmental stages of the host animal. In other words, it is indeed the song, not the singer. You have different microbes soil microbes in different areas. There's a chance invasion of species into the newborn calves. There's differential establishment of the microbial community based on the genetic background. There's niche modifications allowing differential settlement for late colonizers. So the phenotypic outcome, how much methane, for instance, is being produced, how much energy is being produced, depends on the different microbial populations that one gets. And there is a huge amount of research now on how to direct the metabolism towards a less methanogenic phenotype. So you have diet and hosts and what the microbes get and what the cow gets. Their habitat that selects for similar groups of microbes out of available species pool to perform the task, again, the function of degrading and fermenting plant material. However, microbial communities of different ruminant species remain distinct from each other, even in different breeds within a ruminant species. The Norwegian red and the Holstein Friesian cows, for instance, they exhibit broad physiological differences that affect the composition of the microbial community, even under the same conditions. However, there is a core community of microbial species, some of which are found in 100% of the cows surveyed. And those rankings are consistent between cow breeds. These seem to be the keystones of the ecosystem. Ruminococcus, you might imagine, Ruminococcus. We have them in us too, but Ruminococcus flavifaciens is a critical part of this ecosystem. Fibrobacter is another. They're capable of digesting cellulose into soluble sugars. Okay, so this is nutritional symbiosis. The grasses and grains are affordances to the ruminants. The microbes are a crucial and necessary part of the cow. However, what I find fascinating is the sympoiesis, the developmental symbiosis, because the rumen is formed in part by the microbes. As long as they're getting fed milk, the rumen is bypassed. There's a tube here, the esophageal tube, which goes right into the abomasum, so that the abomasum is getting the milk. However, once the cattle is fed grain, that tube disintegrates and the grain goes into the rumen. Now the bacteria in this rudimentary rumen are able to metabolize it into butyric and propionic acid. And what happens is it starts developing. So you have the expansion of the rumen, the elimination of the esophageal groove so that the food goes into the rumen, the development of the rumen itself, its rumen papillary system, and its musculature. And what Sander and company found in 1959 was that they could get this effect by putting butyric acid into the milk that will induce the growth and differentiation of the rumen. More recently, all sorts of transcription factors were found when they added butyric acid into the rumen of the cow. The rumen starts differentiating into a functional rumen. It gets muscles, it gets its epithelium, it gets all these transcription factors, paracrine factors, upon stimulation with butyric acid. So rumen bacteria, which are present since day 2, are now able, once weaning occurs, once they're given grain, to ferment the dry feed into butyric acid. The butyric acid causes developmental changes, generating the mature rumen. The bacteria construct their proliferative niche, the rumen. This is developmental symbiosis. The butyric acid given to unweaned cows in their diet or directly into the rumen will cause these changes. Now, the bacterial community can ferment the grass and grains into simple carbohydrates, which is nutritional symbiosis. So you get developmental symbiosis, you get nutritional symbiosis, all in this evolutionary framework of being able to create a rumen. Developmental symbiosis has opened an evolutionary trajectory. The establishment of symbiosis between potential herbivores, my emphasis, and various microorganisms in the gut may have been such a necessary precondition for the evolution of feeding on such plant tissues. And as Moraes and Mizrahi say, bacteria are obligate inhabitants of the rumen. Without them, the host animal would not survive. There is no such thing as a cow without its symbionts. It's a holobiont. So what I've tried to show is that evolution, metabolism, development are three rivers. The integration of replication and metabolism happens in symbiosis, as Dupré and O'Malley had said. The holobiont is a physiological entity. The holobiont, moreover, is a developmental entity, and I use the gut capillaries, the pancreatic beta cells of zebrafish and sponge metamorphosis as examples, and that the three streams are brought together. And we could see this in invasive beetles and bugs, in metamorphosing sponges, in the dung beetles, and most prominently in the ruminants. So the river flows. Congratulations, John. Thank you.